everyone, my name is Farhat Jahan. I am pursuing my PhD in Botany at ICA National Research Institute, which is situated in Cuttack, Odisha. So today I am going to teach you on the topic cytodifferentiation of Tracheary elements and Sieve elements. So today we will dive into one of the most fascinating processes in plant anatomy, that is cytodifferentiation. Specifically, how it gives rise to two critical vascular tissues that are tracheary elements of xylem and sieve elements of the phloem. These specialized cells are actually essential for water and nutrient transport in plants. Understanding their development gives us insights into how plants grow, how they survive and function efficiently. Let's start with the term cytodifferentiation. So the term cyto means cell, and differentiation refers to the process by which general or unspecialized cells become specialized. So cytodifferentiation altogether means the transformation of an unspecialized cell into a specific functionally active cell. Before we understand how cells differentiate into tracheary or sieve elements, let's revise the basis of what a cell is. So, as we all know, cell is the smallest unit of life capable of performing an essential function. The term was coined by Robert Hooke in 1665. He saw tiny boxes in cork under a microscope. Later on, Anton van Leeuwenhoek in 1674 observed live cells and opened the door into the microbiology. So today what we study, the microbiology, dates back to the discovery of the Anton van Leeuwenhoek. So now cells can be broadly classified into prokaryotic and eukaryotic cells. Prokaryotic are primitive, like bacteria, they don't have true nucleus. Eukaryotic are complex cells with a defined nucleus and membrane-bound organisms that are plant and animal cells. Now let's also understand how plant and animal cells differ. For reference, I have given here the picture of a prokaryotic cell and a eukaryotic cell. So plant cells have a rigid cell wall and chloroplast for photosynthesis. However, animal cells lack this but have structures like lysosomes. This understanding is crucial because the process of cytodifferentiation occurs in the plant only in the eukaryotic cells, and that to the plant cells, which leads to the creation of a complex tissue like xylem and phloem. So now we understand a cell. Let's look at how cells build the entire plant body. So this is the cellular organization. From a single cell to a complete organism, let's look how cells build the entire plant body. All living organisms follow a structural hierarchy, that is, cell constitute the tissue constitute the organ constitute the organ system, and organ system ultimately makes the organisms. It all starts from the cell, that is the smallest structural and functional unit of life, as we all know. When many similar cells work together, they perform a tissue like parenchyma, collenchyma, or vascular tissue. Different tissue combines to form an organ such as a leaf, stem or root. Now the organ starts to work together in a system like the root or shoot system, which together make up the entire plant organism. In this hierarchy, the cytodifferentiation plays a key role in the tissue as well as in the cell level. For example, cell in the vascular ganglion differentiate into specific tissues like xylem or phloem based on their function. So cytodifferentiation is what transforms a simple cell into the unit that constitutes the whole plant structure and performs its important function like transport of water as well as nutrients. Now that we understand how cells form the tissue, let's look at the main types of plant tissues. So the major types of plant tissues are broadly classified into two types, that is meristematic tissue and the permanent tissue. The meristematic tissues are actively dividing cells. They have the ability to divide and redivide. These are mainly found in the root tip, shoot tips and ganglion. These are responsible for growth both in primary as well as in secondary development. Then this includes the apical meristem, lateral meristem and intercalary meristems. These are the starting point of cytodifferentiation. The meristematic tissue is the starting point from where the cytodifferentiation takes place, as they give rise to all other tissues. So the meristematic tissues form the permanent tissue. So the meristematic tissue forms the permanent tissue. So the permanent tissues are non-dividing mature cells that have taken a specific role. That means they have lost their ability to redivide and they already are in that structure which is capable of performing a particular specific roles. It is further divided into two types, that is simple and complex permanent tissue. So that simple permanent tissue is that is made up of one particular type of cell which includes parenchyma, collenchyma and sclerenchyma. Parenchyma is living thin-walled. It helps in storage and photosynthesis. However, the collenchyma is living and thick-cornered, which provides flexibility. The sclerenchyma is known as the dead, very thick-walled, which gives strength to the tissue. So now let's come to the complex tissue. So the complex tissue are of two types, that is xylem and phloem. Xylem includes tracheids, vessels, fiber and parenchyma. Phloem includes xylem tube elements, companion cells, parenchyma and phloem fiber. So xylem conducts the water and minerals and it has tracheids and vessels. However, phloem conducts food from the sieve tubes and companion cells. So in cytodifferentiation, cell from the meristematic tissue differentiate into these specialized permanent tissues which are the xylem and phloem, especially the complex tissues. And the xylem is very important for transporting the water from root to all parts of the plants, and the phloem is important to transport food and involved in photosynthesis from leaf to all parts of the body. Now let's discuss about what is growth and development. As we all know, meristematic cell undergoes growth and development. So growth is the irreversible increase in size, mass or volume of the cell. But development, on the other hand, is a broader term that includes growth, differentiation, morphogenesis. That means overall life processes includes the development. Now let's understand the key terms in the cytodifferentiation. What do we mean? What do we include in cytodifferentiation? First term comes differentiation. It is the process by which unspecialized meristematic cells become specialized, that are tracheary elements or sieve elements. For example, the tracheary elements of xylem and sieve elements of the phloem become specialized. These cells change in structure, function permanently and becomes permanent tissue. From the meristematic tissue, it becomes permanent tissue. Now it comes to de-differentiation. When a mature cell regains the capability to divide, for instance during wound healing or callus formation, parenchymatous cells often do this. Redifferentiation happens when this differentiated cell again specialized into a new type, such as callus regenerating into xylem or phloem during tissue culture. Together, this process allows plants not only to grow and mature but also to repair and adapt. In cytodifferentiation, we also learn a term that is redifferentiation, that is respecialization into new function, that could be secondary phloem or secondary xylem. In cytodifferentiation, we mainly focus on differentiation, but the de-differentiation and redifferentiation are very important and crucial in regeneration and biotechnological applications also. So let's come to our topic, that is cytodifferentiation. So cytodifferentiation is the process by which simple unspecialized cells become specialized for specific functions. In plants, this leads to the formation of the complex tissues like xylem as well as phloem. Here we can say this is the picture of a xylem and this is the picture of the phloem. And the vascular tissue undergoes cytodifferentiation and formation of xylem element, and here the phloem elements. So its definition would be read as process through which unspecialized plant cells, that is meristematic cells, develop into specialized cell with specific function. In conclusion, we can say that the meristematic cell undergoes changes, division and redivision in order to make it a permanent tissue, and which is capable of performing important function and particular specific function, which may be transport of water or transport of fluid. Now, xylem is responsible for transporting water and mineral from the root to the other part of the plant, as I already told. It consists of tracheid vessel elements which are dead, hollow cells and thick. On the other hand, phloem transports food and organic nutrient, mainly sugar, from leaves to other parts of the plant. It is made up of sieve tube elements and companion cells. Unlike xylem, phloem cells are alive and work in coordination to make active transport. So through cytodifferentiation, cells gain the structure and function needed to become either water conducting or food conducting tissues. Now let's focus on the cytodifferentiation of tracheary elements. So the tracheary elements are parts of xylem and help to conduct water in plant. Tracheary element includes tracheids and vessels. Their differentiation from meristematic cells involves several key steps, that is cell elongation. The immature cell increases in size and becomes elongated. The elongated cell may be parenchymatous precursor. Cellulose microfibril arranged longitudinally here. Then the secondary wall thickening takes place. Lignin is deposited in specific patterns like spiral, annular, reticular, scalariform, and pitted form. Then the lignification takes place in these phenomena. The wall becomes heavily lignified, making them rigid and waterproof so that they can easily transport the water. Lignin strengthens the wall resistant to collapse, that makes the cell not to collapse easily, and wall thickening begins at the corner and spread. But first it starts from the corner, then it spreads to the whole overall wall. Then PCD takes place. PCD refers to the programmed cell death. The cell loses its nucleus and cytoplasm, becoming dead hollow tube. Here you can say dead hollow tube is present. So the nucleus degenerates, tonoplast structures, and cytoplasmic contents are digested. The many hormones and transcription factors here works for the cytodifferentiation of the Tracheary elements, that could be auxin and racinosteroids, some signal molecule as well as transcription factors. Here, as we can see in the picture, that this is the mesophyll cell, which has shown here clear nucleus is seen. This is the cytoplasm, this is the chloroplast. First of all, the secondary wall deposition starts, and the vacuole formation has become. The cell has been elongation has taken place, and the secondary wall formation has started from the corner and spread to the wall, and then programmed cell death takes place. All the nucleus degenerates, tonoplast has content has digested, and the plant has become hollow. So this is the picture of the mature tracheary element. So the final cell is now strong, dead, and capable of conducting water efficiently through the plant. This transformation is controlled by plant hormones like auxin and genes, ensuring that the cell develops into a perfect water conducting structure. Hope this you all have been cleared from how cytodifferentiation takes place in making the tracheary elements. Let's move to the next portion, that is cytodifferentiation of sieve elements. So sieve elements include sieve elements includes sieve cells in gymnosperms and sieve tube element in angiosperms. They remain alive but lose their nucleus. So sieve elements are the main conducting cell in phloem. The steps involved in the cytodifferentiation of sieve elements could be underlined in few headings. The first step is cell specialization. First, the mother cell differentiates into sieve cells. Here we can say the mother cell has differentiated into a complete sieve cell. Now become elongate and thin wall. It elongates and becomes thin wall. Formation of sieve areas in plates. Pores from the transverse wall, that are sieve plates, that longitudinal sieve areas facilitate the flow. So the sieve plates are arranged here and it facilitate the flow, and these sieve area later on form a complete sieve plate-like structure. Here we can see the sieve plate-like structure. Selective organelle degenerates or degrades. Nucleus, ribosome, and Golgi body disappear. Retains plasma membrane, endoplasmic reticulum, and mitochondria near sieve pores. So near the sieve pore, only mitochondria, endoplasmic reticulum, and plasma membrane are left. However, Golgi bodies, nucleus, and ribosomes disappear, and the companion cell associate. At last, the companion cell here associate one after another in a chain manner. The sieve tube elements lack nuclei. The sieve tube elements lack nuclei. Depend on metabolically active companion cells. As the lack nuclei, so can they depend on the companion cell. Both cells arise from the same mother cell. From this same mother cell, both these cells arise. That means companion cell as well as the sieve tube plates. The hormones actively participating here are auxin and cytokinin. In case of tracheal elements, what we have learned, auxin and racinosteroid, but in case of sieve elements, here it is auxin and cytokinins. Callose deposition. What do we mean by callose? The group of undifferentiated mass of cell is known as callose. Its deposition also favors the formation of the sieve elements and few transcription factors. Now that we have learned about what is cell, how cell constitute to the organism, how cytodifferentiation takes place, how tracheary elements, sieve elements are made following the steps of cytodifferentiation. To summarize everything we have learned today, we begin by understanding the cell is the basic unit of life with two main types, that is prokaryotic and eukaryotic, how plant cell differ from animal cell. Then we saw how cells form tissue, organ and organ systems. Then we saw that the complete hierarchy, in the complete hierarchy, the meristematic cell where it works and becomes the permanent complex tissues to work certain functions and There we also learned that in the tissue and cell level the cytodifferentiation works. Then we saw how cell types leads to the formation of complex tissues and simple tissues, how xylem and phloem are complex permanent tissue that arise through specialization. What do we mean by cytodifferentiation? It is the process of making unspecialized meristematic cell become specialized vascular cells. We also studied the process of growth, development, differentiation, dedifferentiation and redifferentiation, where we explained to show how cells become specialized or revert when needed. Cytodifferentiation is the central process that transfers undifferentiated cells into functionally distinct ones. In xylem, cytodifferentiation results in dead lignified tracheary element, tracheids and vessels for water conduction. It conducts water from root to all parts through xylem. In phloem, it leads to the formation of Sieve element with companion cells adapted for food transport. This entire process ensures the plant development, efficient transport, allowing them to grow, survive, and adapt. At last but not the least, cytodifferentiation is not just a biological process, it is a key to plant life survival and its function. I hope that this video gives you enough knowledge to understand the cytodifferentiation and how tracheary elements and sieve elements are formed. That's all I can conclude. Thank you all and have a good day.